So Walid Korani is going to be presenting on an efficient means for calling moderate to long structural variations through pairwise genomic comparisons and Kmers. Walid, take it away.
Thank you, Jeff. Hello, everybody. The talk is about calling variance, long to moderate variance between genomes. First, as Josh mentioned, there is plenty of issues to use low coverage short reads to call variance. Some set of this related for the genome itself, the genome probability, peanut, for example, or this our target, main target, is tetrapolybloid. So it's polybloidy make an issue for the duplication between the two genome itself, or where the best location for the reads to be matched, and also the duplication inside genome itself, not just the whole genome duplication. Peanut doesn't have a lot of heterozygosity place, but for some species or some locations that is heterozygous, it make an issue too. Also, some issues related for the sequence itself. Illumina sequence is high accurate, but still have some errors, and by these errors can make many false positive for calling variants. And the main issue for low coverage, for short reads, is calling the indels. Around 10 years or so, there was the calling variance in peanut was very low efficient. Different strategies has been developed, different pipelines has been developed, and for SNPs, for example, it doesn't exceed 10% accuracy, and for example, SSRs, it has around 6% or so. Then Josh and Beggy developed SWEEP, as Josh mentioned, using the structure of two subgenome versus the structure between individuals. And based on that, SNP chip 1 has been developed, and the accuracy raise, or we had a jump here from 10% or less to 40%. Then using the information in the SNP chip 1, machine learning was used to train models to call SNPs, and we had another jump here to 80%, which has been used for creating SNP chip 2. And finally, COFO. With COFO, we could get more than 90%, even with low depth, either for SNPs and for indels. But COFO, because he used just the short reads, so far we could recognize the location of most of the long indels, but we cannot get the structure for them. So a lot of genomes will be available, and we can utilize the availability of that to create the possible, create a pipeline or tool to get all possible insertion, deletion, moderate and long, using this information. This may be helpful for a lot of directions, like creating band genome or whatever. So we create what we call it Cyclops. In Cyclops, we have different layers of filtering to get the high confidence insertion, long to moderate insertion deletion between the two genomes. Let's say compare between a theoretical genome here with 3 chromosomes. So if we need to compare them together, we have to put all possibility to compare each chromosome for one genome to the rest of the genomes. So the first step to get that, we need to create an alignment. But to compare between 2 chromosomes, it's tough to do directly local alignment. So we start with global alignment. Actually, this method is used before using Kmers, and we use it here, but with high efficient, as I discussed later, for memory and threading. The issue of global alignment, the idea of global alignment, you put the two chromosomes together and start to build the matrix from the beginning of the chromosomes. So you have an issue for the gapping location. You have an issue for, you don't have statistical model. You cannot build statistical model to have judgment of the matched region, it's accurate or not. And also it's extensive regarding for computer resources, trading, and for memory. So the first idea of Cyclops, we have a good management for trading. We create C++ script. Actually, we have a serial of the C++ script, what we call batchers. These to handle any programs that doesn't utilize the memory, doesn't utilize threading well, and manage it and use it with high efficiency. For example, in the theoretical genome, so we can split the process in 9 different sub-process, and each one can be run in independent thread, and based on the computer efficiency, computer capacity regarding for the memory and thread, we can apply the program. But when we get a new region, if you compare between chromosome 1 here from one genome and the other, and we get this black region, because global alignment has a lot of issue, as we mentioned, especially around gap locations and statistical model. So we need to fill the gap between the local alignment and the global alignment. So we use Kmers. The default Kmers for Cyclops is 35. This is very efficient length. We get the new regions that has been discovered by the first step by Kmers or global alignment. Then we create serial of Kmers for that. Then we use these Kmers and do local alignments against the opposite genome. And we check the regions that has matched. So this may be coming from the gaps from the first step. So we drop them and using the new region to rebuild back for the insertion location. Actually, we do it in 2 directions to get insertion and deletion. This is a kind of case study has been done for NC 94022 and Tifrunner. With this method, we could extract 300K deletion. Actually, deletion is more than insertion here, and 17K insertion. The length is 40 base pair to 4 KB. If we split them in 3 groups to be easier to visualize, this is the length between 35 to 100 to 200, almost 200. Most of variants here with deletion, small deletion. And this is another set from 200 to 1,000. Again, most of them are deletion. But the last part is the long endels, most of them here was insertion. Again, because this happened in very efficient way for using memory and threading. This happened for comparing the 2 genomes in 2 directions in less than 3 hours. Only Kmers can be done in days and weeks. But this way, this method allows for efficient way for using the memory, using the thread. So this reflects on the time. This one thing. Another thing, we can create a full library between the 2 genomes regarding for long and moderate insertion and deletion. And that's it. Thank you so much.
Excellent. Thank you, Walid, for your presentation. We do actually have time for a question if somebody is eager to ask. I assume that this could be a follow-up to either Josh or Walid. I'm sure Walid could probably answer on behalf of Josh, if Josh is still there. Any questions from the group? If not, I guess I can go back to, I guess if Josh or Walid wants to answer this one from Jianping Wang. Thanks for your talk, Josh. What are the main differences and considerations in terms of the algorithms, algorithms between the sweep and COFU in peanut SNP filtering? Why is COFU more accurate?
Yeah, thanks, Jinping. That's a good question. The way I would answer it is basically to say that in sweep, we were relying on these regions that had pile up, and we did a good job, but then we couldn't control well for the regions that had more than 2 loci pile up there, and so those were sort of, that's what drove the noise when using those pipelines. So with Khufu, we don't do that, right? So we're actually filtering on the read location. So we're filtering how accurate the mapping is of the read. And so we filter out those reads that aren't, they don't map well to that location. So what that does is it now sort of gets rid of the bias that we had before. We have really pretty accurate locations for where the SNPs are, and we're really confident about the reads that map there. And so that's why at the low coverage, it's still very accurate. And it also allows us to see what, like, almost all of the SNPs that are in the population. And so, yeah, that's really the difference between the two.
Walid, anything to add?
No, that's it.
Okay.
Yeah, for sweep, it's regarding for the genome, subgenome structure, but for COFO, mainly for correct location, localizing the reads back, and in addition to different filters for the reads themselves. But generally, we could extract almost all SNPs in the population if they have at least 1x coverage.
Excellent. Excellent.